Hello everyone, welcome to this session. In this session, we are going to study about the mutation breeding. So, what is mutation breeding? High frequencies of chromosome mutations are desirable, right? High chromosome mutations are desirable in the modern breeding techniques. That means nowadays in the breeding technique, there is a requirement of the chromosome mutation. There is a requirement of chromosome mutation. So as they provide new source of variation. So by this chromosomal mutation, there is a variation in the organism. Okay, so that is why this chromosome mutation is required for the formation of new varieties of the crops. Okay, so in the modern breeding techniques, there is a requirement of the chromosome mutation so that we can make different varieties of the crops, new varieties of the crops. Multi-allelic nature of loci in a polyploid has many advantages that are useful in breeding. So in polyploids, one particular gene is present in multi-allelic form. Means for one particular gene, there are numbers of alleles. Okay, in polyploids. We have already discussed a polyploid in a different session. So we are not going to study much about it. But yes, there is a multi-allelic nature of loci in polyploids, and that has a great advantage in the breeding technique. The masking of the deleterious allele means the alleles which are not useful for the plants. So that particular gene get masked. How? So the masking of the deleterious allele that may arise from the induced mutation. Okay, so the deleterious allele, the genes, the alleles of a gene that is not useful for the plant get masked by induced mutation, So by their dominant forms, cushion polyploids from the lethal conditions often associated with the inbreed diploid crops. So this induced mutation mask the deleterious allele, okay, and their dominant forms cushions polyploid from lethal conditions. So this cushions polyploid means they protect them from the getting to the lethal condition. Okay, in this concept has been instrumental in the evolution of polyploids during the bottleneck where there is enforced inbreeding. So this concept has been very, very important in the evolution of polyploids. So how the polyploids are formed? By induced mutation. Mutation breeding exploits the concept of gene redundancy. Gene redundancy means one particular gene, one character, in fact, one character or trait is controlled by number of gene. It is the effect, this trait is the effect of 4 different gene, right? And if one of the gene get inactivated, there is not much effect on the character or the trait. That is called as gene redundancy. Okay, so mutation breeding exploits the concept of gene redundancy and mutation tolerance. Okay, mutation tolerance in polyploid crop improvement in 2 ways. So this polyploid crop can withstand with the mutation. They can tolerate the mutation. Okay, because for one particular gene, there are numbers of alleles. Okay, first, so how they improve? They improve in 2 ways. The crops can be improved in 2 ways. How? The first, polyploids are able to tolerate the deleterious allele modification post mutation. Means after the mutation, these polyploids are able to tolerate the deleterious effects of allele, some of the allele which having the mutation and their effect is very deleterious to the polyploids or to the crop. Okay, so that can be tolerated. And secondly, they have increased mutation frequency because of their large genomes resulting from the duplicated condition of their genes. So when a particular genome is duplicated number of times, the probability of getting mutated increases. This is a sort of mathematical explanation that the probability of genome mutation increases because you have the number of copies of the genome. So the probability of getting mutated also increases in that fold. Okay. So these are the two ways improvement, and while increasing this mutation frequency, there can be, so the crop can improve in two ways. First, the polyploids are diploid. The polyploids are able to tolerate. The crop improvement can be done in two ways. First, the polyploids are able to tolerate the deleterious allele modification. They can tolerate the deleterious effect of some of the allele, right? And they have increased mutation frequency because of their large genome, resulting to the duplicated condition of their genes. So this high mutation frequency observed with the polyploids may be exploited when trying to induce mutation in the diploid cultivars that do not produce enough genetic variation after the mutagenetic treatment. So this approach has been used in mutation breeding the acmens, that is the nut orchids, a type of orchids, by first forming the autotetraploids through the colchicine treatment, followed by the application of first neutron and the X-rays. So what happened? The colchicine treatment, what does it do? It increases the genome copies, right? It increases the genome copies as they form diploid gametes. They form diploid gametes. And we can increase the copies of the whole genome in this way, right? And after that, fast neutron or X-ray treatment can mutate and we can have the mutation breeding. This approach can be used in mutation breeding. In this study, the autotetraploids were found to have 20 to 40 times higher mutation frequency than the corresponding diploid cultivar. If we compare with the diploid crop and one that is autotetraploid crop of the same type, we are having 20 to 40 times higher mutation frequency in the autotetraploids if we compare to the diploid cultivar. Okay. So, mutation occur in two ways in the organism. By alteration in the nuclear DNA, that is called as point mutation. At one particular point, the genome gets mutated. And by chance of cytoplasmic DNA, that is the cytoplasmic mutation. Cytoplasmic DNA also get mutated. So, if that happened, then that is called as cytoplasmic mutation. Spontaneous mutation. Now there is a types of mutation. We have a different types of mutation. The first one is the spontaneous mutation. Mutation occur in the natural population. In the nature also there is mutation. Okay. So when it occur naturally, mutation occur naturally, then this is called as spontaneous mutation. But when mutation may be artificially induced by various mutagenic agents by man, then it is called as induced mutation. Now, induced mutations are of two types. What are those? Macro mutations. Mutation with a distinct morphological change in the phenotype. That brings morphological change. The phenotype gets changed. Okay. So that is the mutation with distinct morphological change, that is called as macro mutations. Micro mutations. Mutation with invisible phenotypic change. You cannot see that change, but yes, there is a change, okay, and that is called as micro mutations. Moving to the types of mutation in this context only. Induce desirable mutation and exploiting them for crop improvement. So induce desirable mutation is used for exploiting for crop improvement. It has been used for the improvement of the crop. So mutation breeding is commonly used in produce traits in crops such as larger seeds. We want to have a crop like rice and grain, like rice and wheat, that their grain should be larger, So they are used for the larger seeds, new colors. We can see here this radish in so many colors, okay, or sweeter fruits. We want our fruits to be more sweeter, right? Then also the mutation breeding is used. That either cannot be found in nature or even be lost during the evolution. So now here you can see this fruit is without any seeds. No seeds are there. Okay. So these types of fruits are preferred, and mutation breeding, that is why commonly used to produce such type of crops. Mutagen, physical or chemical agent which greatly enhance the frequency mutation. So these are called as the mutagen, the chemical agent that is used, means chemical or the so mutagen, physical or chemical agent which greatly enhance the frequency mutation. These are called as the mutagen. And what is the mutant then? It is an individual organism or a new genetic character arising or resulting from the instance of mutation. So once the organism or an individual get mutated and it has a new genetic character, then it is said to be mutant. And what are the agents that we are using? That can be a physical agent and the chemical agent, and that is called as mutagen. So our mutagens are of two different types, physical mutagen and the chemical mutagen. Physical mutagen, ionizing radiation. So that this physical mutagen can be in the form of radiation, and that created ionizing, right? So particulate radiations, alpha rays, beta rays, fast neutrons and thermal neutrons. These are some of the particulate matter that causes radiations, and that induce mutation in the organism. Non-particulate radiations, they do not have any particles, still they produces radiation, and that also induces mutation, that is X-rays and gamma rays. Non-ionizing radiation, that is the second type, ultraviolet radiation, UV radiation. Okay, so that is non-ionizing radiation, and that causes very deleterious mutation in the organism. Now we are having chemical mutagens. Okay, the second type after the physical, that is the chemical mutagens. So alkylating agents, this is a one type of chemical mutagen, causes mutation by adding alkyl group to the various position in the DNA. So this alkyl group is added, and that changes the composition of the DNA material, and that causes mutation. EMS, ethyl methane sulfonate. This is one of the example of alkylating agent. Methyl methane sulfonate, sulfur mustard and nitrogen mustard. These are the different examples of the chemicals that causes mutation, and they comes in the category of alkylating agent. So in the examination, this could be asked that which of the following is not a alkylating agent or alkylating agent, depending upon the type of distractors they have. Acridine dyes, they are very effective mutagens, lead to addition or causes frame shift mutation. This is very deleterious type of mutation. Okay, acridine dyes. Examples of the acridine dyes are proflavin, acridine orange and acridine. Next, in this same category, that is the chemical mutagens, comes the base analog. Chemical composition similar to the DNA bases, right? We are having the different bases in the DNA, nitrogenous bases. So they are similar to those nitrogenous bases like bromouracil, 5-chlorouracil. Okay, these are the two base analog examples of the base analogs. Now other mutagens are like nitrous acid and the sodium azide. Okay, so these are the very important, some of the chemical mutagens you must remember. Okay, so gene mutation, that is also called as the point mutation. Change in the specific sequence of the nucleotides in DNA molecules, leading to the formation of new type of protein or preventing that of the normal protein. Okay, so if this is a sort of a DNA that is having a one particular gene A, right, and it produces a protein. This is a protein and this is the functional one. This is a functional one. Right? Functional protein. Now, after mutation, there is a slight change in this gene. So I am just denoting as A dash. Okay. Now it produces same protein, but this protein is not same as it was produced earlier by gene A. Okay. So this is a non-functional protein. Okay, so change in the specific sequence. So in this particular, there is a change in the specific DNA. So here you can see this is the normal DNA here, right? This particular part, now it get mutated. So whole gene structure get changed, and the trait that is governed by that particular gene also get changed. Chromosomal mutation. Earlier we were talking about the gene. Now we are talking about chromosomal mutation. So deletion or deficiency, loss of the chromosome segment. So here we can have a one particular chromosome, right? And if there is a loss of this particular part, then the size of this chromosome get reduced and this particular gene get lost. Okay, so deletion or deficiency, that is the loss of chromosomal segment. Duplication, doubling of the chromosomal segment. Sometimes the chromosomal segment get doubled. Okay, one particular gene is present here, that is A. Okay, now when it get duplicated, it is present in two position like this. Inversion, rearrangement of the group of genes in a chromosomal in such a way that their order is reversed. Okay, this is called inversion. Order of the gene, suppose the order of the gene is A, B, C, D. Okay, so sometime what happened, there is a reversal, that is called A, B, DC. So this is the effect of inversion. Translocation, change in the position of chromosome or exchange of the chromosome segment. Sometimes one segment, one particular gene of one chromosome get deleted and get attached to some other chromosome or some other chromosome position even. So that is called as translocation. So these are some of the type of chromosomal mutation. Whole chromosome is involved here, right? And that is why it is called as chromosomal mutation. So deletion, duplication, inversion and translocation are the different types of chromosomal mutation. Now, genome mutation, very important. Change in the set of chromosome. All the chromosome get mutated or the chromosome get duplicated. Number of copies of a whole chromosome, then it is called as genome mutation. So here we are having some of the crops that got mutated and it has been used as a mutant variety. So enhancement of some global mutant varieties we are going to discuss here. Okay, the first one is about the rice. The impact of induced rice mutants in applied research is the best by the development of improved rice varieties through mutation. So mutation has been used to develop a new varieties or the improved rice varieties. Okay, so this is very, very important. And that particular rice which we develop by the mutation process is called as induced rice mutants. During the past 5 decades, more than 800 varieties of rice has been developed across the globe. Okay, so either directly from the induced mutation or as a result of crossing such as mutants with other breeding lines. So now you can understand we have different varieties and how these varieties, so many varieties of rice, how it can be developed. Afterward, the semi-dwarf mutant Rami was released in Japan, which have significantly increased yield because of their lodging resistance. So now this is the name of the dwarf mutant Rami was released in Japan. This is also one of the mutant varieties of the rice. Kalros 76 and Basmati 370, semi-dwarf varieties of rice with a short end straw, has revolutionized the rice production in USA and Pakistan respectively. So these are some of the varieties of the rice that has revolutionized the rice production, especially in USA and Pakistan. The first rice varieties, that is KT 2074 and SH 3021, developed through mutation that were released in China in 1957. Now, developing drought salinity tolerance in the wheat crop. Now we will talk about some of the mutant varieties of the wheat crop. Sharbati Sonara, a semi-dwarf non-lodging mutant variety, has a significant contribution to the wheat production in India. This is one of the varieties, mutant varieties of the India that is very, very famous. Sharbati Sonara was produced from the red-grained Mexican variety, that is 60, Mexican variety 60, by gamma irradiation at ICAR, New Delhi, India. So Indian Agriculture Research Institute has developed this Sharbati Sonara variety of the wheat. A high yielding mutant Stadler developed in Missouri, USA, had to left rust and loose smut. These are the two very common diseases that occur in the wheat. Okay, so they can tolerate better logging resistance and early maturity. So this particular Stadler, mutant Stadler, can withstand with the loose smut and leaf smut because they are having better lodging resistance and early maturity of the crop. In Italy, durum wheat cultivation area was significantly expanded due to the cold tolerant and the mutant varieties. Mutation breeding has been greatly successful used the breeding barley. Now we will talk about some of the lodging resistance in the barley crop. Okay, so mutation breeding has been very successfully used in the breeding barley also. Okay, so the introduction of the diamond and the golden promise, a gamma ray induced semi-dwarf mutant revolutionized brewing industry in Europe. That is the induced barley crop, based on the induced barley crop. Luther gamma ray induced mutant had 20% increased yield. Okay, and the higher end the lodging resistance, and the Pennard had the winter hardiness, better lodging resistance and early ripening. So these are some of the varieties of the barley crop that has been developed, as they have many improvement, as they have better lodging resistance and early ripening. Developing early maturity varieties of the peanut. So there are some other varieties of the crops that has been developed from the mutation, that is developing early maturing varieties of the peanuts, high yielding and the wilt disease resistant chickpea mutants, improvement in the ornamental plants. Some of the ornamental plants also got improved with the mutations. Major economic successes in USA for rice, barley, sunflower, grapefruits, and in Pakistan, that is for the cotton. In India, black gram, Australia and Canada, linseed, and Japan and China, that is for rice. So this is all about the mutation breeding. We will meet with one more interesting session. Till then, thank you very much.